Welcome to the Science and Medicine lecture series. This is called the Junior Investigator Talk or the Young Investigator.
No.
New Investigator. Once again, I have people who can correct me constantly. Okay. In order to give Whitney the introduction she deserves, Lisa Frankel will actually introduce Whitney. Thank you.
Hi all, I feel very privileged to introduce the newest and the youngest member of our division. And we're not only pleased to have Whitney because she looks fantastic on paper, which of course she does, or she would not have been selected for this award, but because she is one of the most inquisitive, hardworking, excited about life individuals and is a real joy to be around. I've worked with her both clinically and on research topics, and in both areas she excels beyond all. And so on paper, just for those of you who have not seen her CV, her family of course knows all this, Whitney is a Washingtonian. She left to go to Harvard to study neurobiology as an undergraduate. And then she returned to her senses and came back to Washington, where she entered medical school and an MD PhD program. She studied malaria as part of the pathobiology department, and then she stayed on for a pediatric residency. And then we were so happy you decided to continue with us as a fellow. And during her fellowship, she branched out into a huge area. The mother host, the infant, and then how the interface, the placental interface, is modified by malaria, and then the passage of maternal and infant cells, how that modulates immunity, or chimeras. So we're extremely glad that you got this award since you're so deserving, and we look forward to your talk. Dr. Harrington.
Thank you all for coming, and thank you for this honor. I really appreciate the opportunity to be able to tell you about my research program. So as Lisa had mentioned, my interest is really in infection during pregnancy and how that shapes the development of the fetal and infant immunity. And as my training is in malaria, I think malaria is a really nice example of this interaction. So when a pregnant woman is bitten by a mosquito, the parasite traffics to her liver. It goes through the liver stage, which is about a week, and this is Plasmodium falciparum specifically. It erupts from the liver stage, and then it circulates in the red blood cells until it reaches the placenta. And when it reaches the placenta here, it will sequester in the placenta. And when we look at that, it's quite interesting. So this is a cross-section of the placenta, and this green cell here is the syncytial trophoblast. So that's that cellular layer that's separating the maternal and the fetal circulations. And these infected red blood cells go into this maternal intervillous space, and they cause a lot of inflammation. So maternal lymphocytes will traffic into that space and fill that up. And as you might imagine, that leads to a number of poor outcomes for that fetus. In addition, we know that when malaria is sitting against the placental interface, it alters the transfer of normal protective immunity. So quite a bit of work has been done on the transfer of antibodies that occurs across the placenta, and relatively less has been done on these rare maternal cells called microchimerism that traffic between that pregnant woman and her fetus. The last thing we know is that although congenital malaria is relatively rare, meaning the actual infection of the fetus, if we look at cord blood from malaria endemic settings, we can find a large amount of reactivity, suggesting that that fetus can be exposed to malaria antigens in utero and mount a tolerant response, meaning it learns to think that that parasite antigen is akin to self. So it's estimated that these effects together lead to 10,000 maternal deaths per year, 1 to 200,000 stillbirths, which is really underappreciated, I think, when we think about global mortality and morbidity, where malaria is really a leading cause of stillbirth. And then up to 200,000 infant deaths that are specifically related to the exposure of the mother during the pregnancy. So this is on top of the additional 500,000 infant deaths that we see related to malaria. So my lab is focused on these rare maternal cells called maternal microchimerism. So what are they? So this slide is summarizing about 100 years of literature. This was initially described in cattle twinning and has since been described in many different animal species. But what we know is that during the pregnancy, rare cells will traffic from this pregnant woman across the placenta and end up in her fetus. Now we can detect these cells distributed really throughout the fetus and throughout the neonate. And then as that neonate ages and becomes a child, we can continue to detect these, and they're present really all over the body. When we and others have looked for these in different organs, we find them essentially in every organ that we've looked. So we can find these maternal legacy cells in brain, in heart, in bone marrow, in spleen, in pancreas, really everywhere. And what's really striking about this is that as the fetus acquires these maternal cells, it becomes tolerant to all of the non-inherited maternal antigens. In other words, all of the proteins that that maternal cell expresses that were not inherited, where that DNA was not inherited by the fetus. So this is really this idea of expanded self, right? Where this fetus learns to understand that these maternal cells are part of itself. Now these cells end up in some really interesting places. So they end up in the thymus, in the blood, in the lymph node, and in the bone marrow. And we think that's quite provocative in terms of their potential role for affecting the development of immunity. In addition to maternal microchimerism, the level I meant to tell you was 1 in 10,000 to 1 in 100,000. So these are quite rare when we look in peripheral blood, but they're also very similar to the level that we see, for example, an antigen-specific T-cell clone, which we know can have very potent reactivity when it's stimulated with the correct antigen. So in addition to maternal microchimerism, which is predominantly what we'll talk about today, I just wanted to call attention to all the other possible kinds of microchimerism. So these are naturally acquired microchimerism. These are not transplant-related or transfusion-related chimerisms. So specifically during pregnancy, as a consequence of pregnancy, that mother can receive fetal microchimerism from her fetus during that pregnancy. We also know that in twin pregnancies, there's an exchange of cells that occurs between those twins. And then there is really a concept in the literature that a first pregnancy can contribute cells to a pregnant woman that then could be trafficked potentially into a second pregnancy. So it really is this interesting idea of these transgenerational interactions between cells. So there are two principal ways that we will measure microchimerism. The first is really a DNA-based assay. So what we do is we identify a non-inherited, non-shared allele. And most of these are at class 2, at HLA class 2, because it's quite polymorphic. We also have a number of other insertion-deletion mutations that allow us to identify any donor and recipient, or most donors and recipients. We will then do qPCR that targets this non-inherited maternal specific allele. So for example, if this is a mom and this is her baby, this haplotype here in the baby is the inherited maternal haplotype. This haplotype here is the inherited paternal haplotype, and this is the non-inherited maternal haplotype. So we will go into this haplotype and we'll look for any non-shared allele here. So for example, this yellow allele or this purple allele, and that can be the target of our qPCR assay. When we find amplification of that allele in the background of the baby, we assume that that DNA came from a maternal cell. Now, really everything I know about maternal microchimerism is from Lee Nelson, who's one of the investigators at the Hutch. And what I've tried to do throughout this talk is acknowledge people along the way, so you won't have acknowledgement fatigue. And so you'll see a lot of pictures of people. The other person who's really contributed a lot to this work in our lab is Sunny Kanan, who was a postdoc in Lee's lab at the same time that I was there, and continues to develop these assays that our lab relies upon. Now the other way that we will detect maternal microchimerism is by looking at whole cells. So this is fluorescence in situ hybridization, or FISH, and we can use this approach in any setting in which there's a male offspring. So if we label the Y chromosome with a green probe here, this is a male who has scleroderma, and this is a piece of lung tissue from that man. And we label the X chromosome with a red probe, and sitting in the middle of this tissue section, you can see this XX cell here. And we presume that that cell is a maternal origin cell. When we go into our malaria-infected placenta, this green line here is the syncytial trophoblast. So again, that's that barrier between the maternal and the fetal circulations. Over here in the intervillous space, we can see a maternal cell where we expect it to be located. And then here we see this XX maternal cell that appears to be sitting within the fetal villi. So it's sitting within that fetal portion of the placenta. So for those of you who don't think about placentas every day like I do, this is a human placenta. It is a fetal-derived organ, so it's really incredibly fascinating. Right? It's grown by the fetus as a way of providing nutrition and exchanging oxygen and waste products between the maternal and the fetal circulation. It's delivered about 15 minutes after the baby is delivered, so baby comes out and then the placenta will separate from the wall of the uterus. If you took this placenta here and you cross-sectioned it, you would see something that looks like this. So if my elbow is the fetus and my arm is the umbilical cord, then these are called the villi. So it's this tree-like projection that projects into this placenta here, and this is where the fetal capillaries sit. So they sit within these different villi, and they're bathed in this low-pressure, continuous flow of maternal blood. And that maternal blood is supplied by these transformed spiral arteries that sit in the wall of the maternal uterus. So if you took those villi and you cross-sectioned them, you would see something like this at term. So this is the third trimester villus in cross-section. And what you can see is that the maternal cells would be sitting out here on the white side. And then if you move through these layers, you see the syncytial trophoblast, which is that cellular layer that's providing that interaction between this mom and fetus. That's a multinucleated cell. It sits against these residual cytotrophoblast cells in a basement membrane. And then within the fetal villi are the fetal capillaries. And that's where fetal blood is flowing. It's inside of these capillaries. So the point here is really that these two circulations are operating in parallel. Right? There shouldn't be direct communication between the two circulations. And anything that's exchanged between the two parties has to occur across these cells. There are really two potential mechanisms of the transfer of microchimerism. The first is really a micro or macro transfusion event. So in this model, what we essentially propose is that there's a disruption in this interface here, and that allows for direct communication between the circulations. Now the intervillous space has a blood pressure of around 10 millimeters of mercury, so that's the maternal side of the placenta. But the fetal villus arterial capillaries, which are sitting inside the villi, have a blood pressure of around 30 millimeters of mercury. So that would suggest that anytime you had a direct disruption between this membrane, you should have a fetal to maternal transfusion event rather than a maternal to fetal transfusion event. And that's what we see with RH sensitization, for example. The alternative model is one of active cell trafficking. And the best data we have from this come from a rat model, where investigators use these human cells, and they show that the trafficking of these cells across the rat placenta required two steps. The first was integrin-mediated adherence to the syncytial trophoblast, and then the second was a VEGF gradient between the maternal and fetal compartment, so the VEGF level was higher in the fetal compartment. And I should just say that I think the main caveat to this study is that the rodent placenta is really structurally quite different from the human placenta. So what does this have to do with malaria? Well, I had alluded to the fact that when we have placental malaria, this intervillous space becomes full of these infected erythrocytes. So each of these red dots with the dark spot in the middle is an infected, is an erythrocyte with a parasite inside of it. And then across that syncytial trophoblast in the fetal portion of the placenta are these uninfected red blood cells. Now when that infection occurs, maternal lymphocytes will enter the placenta and fill up this intervillous space. And in the worst cases of this, it's deemed chronic intervillusitis, and that's really where the entire intervillous space is occluded by maternal mononuclear cells. And as you might imagine, that's associated with quite a number of poor outcomes for this fetus, right? So intrauterine growth retardation, stillbirth, preterm delivery. When we look at specific markers historically of what these cells are, they tend to stain as monocytes or macrophages, although more recent work would suggest that there's also a lot of B cells that are sitting in the intervillous space. Now the last important piece of data is that placental malaria is also associated with an alteration in the maternal-fetal VEGF dynamic. So SFLT1 is a very interesting molecule. It's a soluble receptor of VEGF, and it's a very potent antagonist of VEGF. And it's been best studied as a marker of preeclampsia, and that's new onset hypertension during a pregnancy. And what we think happens is that when the placenta becomes hypoxic, it starts secreting this molecule SFLT1, And that binds maternal VEGF, which results in an increased blood pressure in the pregnant woman and more perfusion to the placenta. Fortunately, it's pretty bad for her. And it turns out that when we see placental malaria, we see something that looks quite like preeclampsia, and even the placentas start to behave like they're preeclamptic placentas. So what we're seeing here are images of the syncytial trophoblast here in red, and this is a placental malaria positive placenta. And green is the S-split, so everywhere that's yellow is that syncytial trophoblast that's producing S-split. When we have a combination of placental malaria and hypertension together, that looks really quite bad. So the first question that we wanted to understand is pretty simple, and that was, in the setting of infection and inflammation in the placenta, does the offspring acquire more of these maternal cells? So to answer this question, we went back to a cohort from Tanzania from the early 2000s, and we measured maternal microchimerism in the cord blood. So that's what we're looking at here on the y-axis. We stratified that by the placental malaria status of the pregnant woman. So the white bars are no placental malaria, the gray are placental malaria without inflammation, and the black bars are inflammatory placental malaria. And we did this because we wanted to understand the difference between infection and inflammation. And when we did this, what we found is that there was a higher prevalence of maternal microchimerism in the inflammatory placental malaria group and in the placental malaria without inflammation group, suggesting that there was a combinatorial effect of infection and inflammation here. And that this was most pronounced in the offspring of first and second time women. And we think that's because they have a really distinct form of placental malaria that's associated with a lot of pathology and chronic infection. This work was done in conjunction with Patrick Duffy and McCall Fried, who were formerly here in Seattle and are now at NIH. Now, because we do this as a quantitative assay with qPCR, we can also look at the level of maternal microchimerism in addition to the prevalence. And when we do this, we see something quite striking. So in our no placental malaria group, the level is 4 per 10 to the 5th, which is very typical for what's described in the literature. By comparison, in our PM no inflammation group, the level is 0.8%, and in our inflammatory placental malaria group, it's almost 2%, meaning 2% on average of the cord blood is of maternal origin. I should note that you might notice these are quite non-normally distributed statistics, so the way that we do this analysis is with a negative binomial model, which is really a count statistic. And that what you're looking at here is a detection rate ratio. So it's saying that for every 1 cell we detect in this group, we detect 200 cells in this group. We had 2 women who had that massive intravillositis, and those 2 women had, when averaged, about 9% of the cord blood looking like it was of maternal origin. So we wanted to understand if malaria was specifically modifying this VEGF dynamic in terms of the transfer of these cells. And as I had alluded to before, the model that exists from rats is that it has to do with more VEGF in the fetus relative to the pregnant woman. And we've hypothesized that as this placenta that's infected starts to produce S-split1 and secrete S-split1, that would bind some of that free VEGF in the pregnant woman and accentuate this gradient between these 2 compartments. So we looked to see if maternal S-split was a predictor of the transfer or level of maternal microchimerism. And we found that in those babies who we couldn't detect maternal microchimerism, all of their moms had really a normal level of S-split, whereas those that were positive had elevated S-splits, and there seemed to be a linear association between these 2. So now that we had described all of these maternal cells ending up in the cord blood, we really wanted to understand what the implication of those cells was. So were they able to actually influence outcomes in these children? And because this was really a malaria model, we wanted to ask specifically about malaria susceptibility. So these kids were followed for 3.5 years after they were born, the children that were born to these pregnant women. And when they came in every 2 to 4 weeks and had blood smears taken so we could tell whether or not they had malaria infection during those events. And what we're looking at here is by age of the child, the probability that, for example, all of the kids who were 50 weeks old, what was the probability that they had a positive blood smear when they walked into the clinic. And what you can see stratified by detection of microchimerism. What you can see here, the dashed line are the babies who were born where we could easily detect this maternal microchimerism in the cord blood. So on average, they had really twice as many malaria infections over time relative to those where we could not detect microchimerism. We thought this was really interesting, and we wanted to ask a follow-up conditional question, which is, will they have infection, but are they actually sick when they're infected? And what we found was quite surprising. So we found that despite the fact that they were getting twice as many infections, when they were actually infected, they were half as likely to be sick from those infections, really dissociating infection from disease. And in malaria specifically, we know that a lot of the pathology is immune mediated. So if you can develop a phenotype in which perhaps you have more infection, but you get earlier protection from disease, that may actually be advantageous in the long run. So those observations really led to the foundation of my lab, which has only been open for a year. And we really are seeking to answer 3 very simple questions. What determines the amount of maternal cells any given child receives? What determines the type of maternal cells they receive? And what are the consequences of this graft, if any? So to answer these questions, we realized that we needed more samples. So we developed a couple of new collaborations and cohorts. This was with Patrick and McCall, but this is a new cohort from southern Mali and Lesabubu, Mali. And this was a cohort in which pregnant women were enrolled around 20 weeks gestation. So in addition to information about placental malaria, we have information about infection throughout the pregnancy. We also developed a collaboration with Maggie Whitney, looking at a cohort that was conducted in Chororo, Uganda, in which women were randomized to 2 different anti-malarials during the pregnancy, and we are using the samples from really the control arm of that trial. After we started working on malaria, we decided that we really wanted to ask this question about other causes of infection and inflammation during pregnancy, and that led us to develop a collaboration with Heather Jaspin and Clyde Gray in South Africa looking at HIV, because we know that HIV also causes systemic immune dysregulation in pregnant women and abnormal placentas. And then after we had done all of that work to develop all of these projects, we realized that we didn't understand normal. So in order to really be able to contrast and compare these different cohorts and physiologic states, we also needed to have a normal pregnancy cohort. And that's what we've been developing here in collaboration with Swati Shri and Steve McCartney at the UW, looking at just normal pregnancies that are delivered every year. So the first thing we wanted to understand was what is predicting the transfer of these cells, what's causing them to traffic into the fetus. So to do that, we had to remeasure microchimerism again from this new cohort, and this is the data from Mali. So every time we do this, we do a unique qPCR for every single individual. And what we found from Mali, which is quite interesting because we had the data on the exposure throughout the pregnancy, was actually that it was 3rd trimester peripheral malaria that was most strongly predicting the amount of microchimerism that these babies were acquiring. But we were happy about this data because it really recapitulated partially what we had seen in Tanzania and made us feel like this was a worthwhile exercise. So then we wanted to test specifically what cytokines or chemokines are associated with the transfer of these cells. So we put together this Luminex panel that Young Ho, who's a research scientist in my lab, ran on Friday. He finished generating this data. So we had plasma samples from these women from mid-2nd trimester, mid-3rd trimester delivery, placental and cord blood. And what we're doing is looking at this panel of 30 different chemokines and cytokines. We're most interested in SLIT1 and then VEGF because of the prior data that we have, but we also want to understand more generally the other distribution of these cytokines. So we've measured this in all of these different levels as of Friday, and we're hot on analyzing that data, so stay tuned for that. Now, as I had alluded to, we also know that HIV causes a lot of placental abnormalities. So specifically, we see abnormal villus morphology, we see activation of the fetal macrophages, which are called Huffbauer cells, and the CD8 infiltration that had been described by other people. So when we went into the cohorts from South Africa, which are cohorts of HIV-infected women and controls, and then of the HIV-infected women, we have what we call HIV-exposed babies. So the babies don't have HIV, but their mothers had HIV during the pregnancy. And what our collaborators noticed, and this is work that was done by Nadia Ikumi in South Africa, is that she can find these CD8 cells that are infiltrating the fetal villi, and they really shouldn't be there. And when she dissects these placentas and extracts the total cells here, she finds that there are far more of these CD8 cells in HIV-positive women compared to HIV-negative women. So we're super interested in pursuing this for a number of reasons. So the first thing that we want to do, and this is work being done by my postdoc who's based in South Africa, Christina Bali, is to go and measure microchimerism in HIV-exposed and HIV-uninfected babies. So she's actively working on that. And then we put in a grant on Tuesday to actually try to see if these CD8s that are infiltrating the villi are of maternal origin. Now the second thing we want to do is phenotype these maternal cells. So we want to be able to say, are they T cells, are they B cells, are they NK cells, are they other APCs, what are they doing? So the data we had to base our initial work comes from SEMI, and this is a paper that we published in 2017 in which SEMI took 27 healthy cord bloods from the hutch, and he bulk sorted them by flow cytometry into memory T cells, naive T cells, B cells, NK cells, and monocytes, and then he measured the microchimerism in each of those subsets. And what he found was that he could easily detect microchimerism in each of these individual subsets, but that the levels were consistently highest in the memory T cell subset. When you look at this data by individual, so each one of these circles represents a cord blood. The size of the circle represents the total amount of microchimerism that cord blood had. You can see the distribution of cell types within each of those cord bloods. So the people who had very large amounts of microchimerism looked like they had a transfusion event from their mothers, whereas other people had a more skewed distribution of, for example, just T cells or just monocytes, although recognizing, of course, that there's some limit of detection of our assay. So based off of this, we realized that we really wanted to be able to isolate these maternal cells, and we wanted to do functional analysis on them, and that was going to require being able to isolate them using flow cytometry. So I moved into Marion Pepper's lab at that point in my fellowship, and we developed a panel, a flow cytometry panel, that we could apply to both cord bloods and paired samples from the pregnant women during that pregnancy in order to describe and phenotype these cells. So it's a pretty basic panel. It's looking at B cells, T cells, different APCs, activation markers. It's looking at memory status, progenitor cells. And here, and we started to test this panel in just normal pairs of moms and cord bloods, just to show that this works. And we see things that are very expected. So for example, in pregnant women, this is looking at B cell memory phenotype. We see a large proportion of B cells that are co-expressing CD27 and 21, suggesting a memory phenotype. In the cord blood, of course, we don't see those cells because we don't expect to see a lot of memory T cells in cord blood. This is Marion, who I spent the last 2 years of my fellowship working with. Now you might notice that in this flow cytometry panel, there are 2 slots that we keep open for HLA class 1 antibodies. So what are we doing with those antibodies? So HLA class 1, unlike class 2, is expressed on nearly every cell in the body, and that is a really convenient tool for us in terms of being able to distinguish donors and recipients. So in this example, you can take this technique and apply it to really any donor-recipient pair. You could use it for maternal microchimerism, for fetal microchimerism. But for the purposes of this exercise, let's imagine that this is a rare maternal cell that's sitting in a fetus. So this maternal cell is expressing A9 and A3. Her fetus has inherited A3 from her, but A1 from its father. So if we use an antibody that specifically tags A9 and conjugated to one particular color, that allows us to uniquely identify those maternal cells. Now this works decently well, but it works even better if we have a second antibody where we can uniquely tag that paternal antigen. And that will allow us to really accurately dissociate these 2 groups of cells from each other. Now there are a number of commercially available antibodies which give us decent coverage. I wouldn't say they give us incredible coverage, but they allow us to look at a number of cord bloods using this strategy, or maternal bloods. And Young Ho has spent a tremendous amount of time validating this approach and really describing the sensitivity and specificity of the approach. So he does experiments where he takes, for example, A9 dominant populations and spikes in these tiny, tiny, tiny amounts of A2 heterozygote peripheral blood mononuclear cells to really mimic microchimerism. And when he goes to a level of 0.1%, which is 1,000 per million cells, you can see that this beautiful population separates that are A2 positive and A9 negative, right? And we feel very good about the way that these plots are looking. You see this smear of what you might first interpret as false positives, right? And that's the point of the double labeling, is to exclude these double positive cells, which we would typically call false positives. Now when we go back and we backdate on these are monocytes, and very interesting to us because we don't know if they're fetal monocytes that have bound maternal antibody or maternal monocytes that have bound fetal antibody. So more on these to come. Our limit of detection we estimate to be around 100 per million cells. So below 100 per million cells, it's very hard for us to tell background from real positive signal. So that's where we tend to stop using this approach. But even here, you can see that we are able to find this minor population here. The other way that we're hoping to mark cells of maternal origin is using bar body labeling. And this is a strategy that will work in any male infant. So in this scenario, there's no unique HLA to mark this maternal cell. It turns out this mom and this baby are HLA identical, right? That's really too bad. But luckily for us, male cells, which would be fetal cells, are XY, right? And these maternal cells have one X chromosome that's being actively transcribed and one bar body, which is inactivated X chromosome. And that allows for a dose response between males and females from X chromosome genes. But when this bar body is repressed, it's repressed with a long non-coding RNA called exist. And we can take advantage of that to fluorescently label that RNA. This is an assay that's published, and it looks like this. So you can actually visualize these bar bodies, and it's meant to be used in cell culture and on slides. And Jackson Stewart, who's my rotating graduate student from pathobiology this quarter, is actively working on adapting this to flow cytometry to be able to use this to accurately call maternal cells by flow cytometry. So back to dual antibody labeling. So this is real data. So this is a mom who actually had preeclampsia during the pregnancy, and this is her peripheral blood that was collected a few hours before delivery, so baby hasn't been born yet. And what we see here is that the mom is A9 positive and A2 negative, her baby is AG positive and A9 negative. So that allows us to really easily separate these populations. And we see this beautiful separation of this minor population of cells that's sitting right here that we're very convinced are fetal origin cells. So we think these are fetal microchimeric cells. And then we see this intermediate population of pesky monocytes that are sitting here. When we tightly gate these cells, we find that not including these, the level of fetal microchimerism is 0.034% in this particular individual, so around 300 per million cells. And then in a totally blinded fashion, John Hauk, who's a technician in my lab, did qPCR to measure fetal microchimerism in the same sample and found a level of 0.037%. So this really delighted us. Really, as if we use this as the gold standard, this suggests that this flow-based approach is actually relatively accurate in terms of dissociating these populations. Now, I told you that we do this antibody labeling in conjunction with this larger panel, so now we can actually look at these rare fetal microchimeric cells and say, what are the phenotype of these cells? So in this particular person, if we first look at the distribution of maternal cells, and for the moment we're ignoring monocytes because it's hard for us to tell in the microchimeric population what the origin of those monocytes are. So this is non-monocytes. If we look at the distribution of cells in the mom, we can see she has a lot of antigen-experienced cells, as we would expect. B cells, NK cells, very typical looking distribution. In parallel, we look at these as cords of maternal bloods together, so we can compare these two sets of data. And when I say fetus, this is cord blood. And here we see also something that would look very typical for a cord blood. So very few antigen-experienced cells, many naive T cells. We see NK cells, which are quite a few, which is described in the literature, and then a nice healthy chunk of CD34 positive stem cells, or progenitor cells. When we look at what the fetus has given the mother, this is what the distribution looks like. So here's the fetal microchimerism from this particular pair, and what we see is that it's predominantly T cells, which is very interesting, so more than 3/4 of these cells are T cells, and a fair number of what look to be antigen-experienced cells. We also see a rare proportion of progenitor cells and kind of a smattering of other things that are distributed here. But the distribution is really not identical between the donor and the recipient, and that's very interesting to us. So using this approach, we'll now go back to our malaria-infected cohorts and the rest of our U.S. cord bloods and try to understand whether different fetuses end up with different distributions of maternal cells and different women end up with different distribution of fetal cells based off of that pregnancy experience. So the next thing we wanted to do was actually sort these individual cells and look at single-cell RNA sequencing to specifically ask a number of really interesting questions. So this is an experiment that Young Ho and John just did in this epic couple of days in which they took 2 million maternal cells and they sorted out 600 fetal microchimeric cells from that pregnant mom. They then spiked back in 3,000 maternal cells. So now we have a sample that's 80% mom and 20% fetus, fetal microchimerism, but they all came from the same pot to begin with. We also ran that in conjunction with a cord blood that's non-sorted, and then our mock-sorted cord blood, and then an unsorted mom. And setting up this experiment, the goals are really to answer a couple of questions. So one, can we correctly identify the origin of the cell? So can we accurately tell on this transcript level what was the maternal cell and what was the fetal cell? Can we figure out if those monocytes are maternal or fetal, for example? Can we confirm the phenotype of the donor cells based off of what we found from our flow cytometry data? A really interesting question that I'm super excited to understand, which is, does a donor cell transform when it enters a recipient? So for example, if you have a bunch of naive T cells and they're fetal and they're sitting in the fetus, they're not doing anything. But when those naive T cells go into a maternal body, do they change their activation status? And that becomes really interesting because, for example, we know that those naive T cells are a predictor of graft versus host disease in cord blood transplant patients because they're poised to react to antigen. And so they're actually interesting to us even though they're naive. And then finally, can we gain clues on antigen specificity? So in the context of malaria and HIV, can we actually sequence T cells and VDJ segments on these individual B and T cells, and then using published algorithms predict what the antigen is associated with those T and B cells? This is also interesting in our normal pregnancies when we want to ask questions about maternal-fetal reactivity in addition. So to demonstrate the two approaches we'll use, this is a computational experiment we ran recently in which we took published 10x single-cell sequencing data, and we looked at the expression of 2 different markers. So I should say these were blinded, so we don't actually know the sex of these 2 different donors, but we can call this donor 1 and donor 2, and we looked at exist expression, so that's that bar body controlling long non-coding RNA, and RPS4Y1, which is a ribosomal protein that's encoded on the Y chromosome. And you can see that we think this is a female donor because 67% of the cells are expressing exist. We can easily find that transcript. 2 of them are expressing this, which is probably actually from a homologous gene on the X chromosome. When we look in this donor, in contrast, none of the cells have exist expression, and 91% are expressing this RPS4Y1. So we think in any male offspring, this approach will allow us to distinguish the cell of origin. The other thing that we're working on doing with the scientific computing group at Children's now is using SNPs to make these calls. So this is not based on the sex of the infant, right? We could do any donor-recipient pair. And what we do here, this is again a computational experiment we ran where we took published data, and this was 3 people who had bone marrow samples done with individual RNA-seq at a depth of 50,000 reads per cell. And then we looked at the distribution of SNPs and saw whether or not we could predict the origin of 2 individual cells from each of these people. And what we found is that indeed the 2 cells from donor A clustered, the 2 cells from donor B clustered, and the 2 cells from donor M clustered together. Now this is not a perfect experiment because these people are not related to each other, right? And in our pairs, in our maternal infant pairs, they share half of their DNA. So it will be more challenging for us, but this is the experiment that we're hoping to run once our sequencing is finished. So it's actively, the machines are chugging in New Jersey right now, and we should have that sequencing data next week. This is Jason Lindner, who's been working on this project with us. So the last question we want to ask is really the functional consequence of this graft. So within the context of malaria, we think there are 2 alternative hypotheses. So the first is that the fetus has actually acquired a maternal cell with memory. So this idea of non-Mendelian heritable memory. This is a malaria-specific T or B cell that this fetus acquired. So to answer this, what we'll do is utilize our sorting techniques. And what people have described in the cord blood in malaria-endemic regions is that you see this malaria-specific proliferation. So if you stimulate with malaria antigens or with whole parasite lysate, you'll see proliferation. And that's always been presumed to be of fetal origin. And our question is really, well, maybe that's actually maternal cells that are antigen-specific and are proliferating. So we'll sort these 2 populations from each other, the mom and the fetal cells, and then we can do an in vitro experiment, where we culture these cells, we stimulate them with parasite antigens, and we ask a very simple question, which is who's responding, who's proliferating, who's producing cytokines. And as we were designing this experiment, by the way, if anyone knows any talented postdocs who'd like to take this on as a project, let me know. As we were designing this experiment, it occurred to us that this was actually an experiment that had already been done in vivo. So when we went back to our malaria cohorts, we realized that for every single one of these children, we have the following information. So this is one particular child from Tanzania, and each of these dots is a visit date. It's a date that they came into the clinic and they had a blood smear taken. They either had parasites or they didn't have parasites. This is data from a single child. So if our hypothesis is that the fetus has acquired, that the infant has acquired antigen-specific maternal cells, when that infant is infected with malaria, those cells should proliferate, right? They should expand. And so what we can do is actually use a child as their own control. So we identify the first parasitemia in this child, and then we find the immediately preceding visit in which they didn't have malaria. And we're able to measure microchimerism in those two samples and compare them together, and that's actually the natural experiment that matches our in vitro experiment. We can, of course, look at later time points where the same thing happens, but in these later time points, we expect that we may see proliferation of both infant and maternal cells. So it'll be a little bit harder to understand what's happening in those conditions. So we're now about to launch this project with Presh Jagannathan, who's now at Stanford. He was formerly at UCSF with Maggie, and actually pull all of the filter papers from all of these kids at their first parasitemic visit, and then pull the immediate preceding cord blood, infant blood, and be able to do this comparison. Now the alternative explanation is that these fetuses are not receiving antigen-specific cells, but that the presence of these maternal cells or this maternal antigen is modifying the way that the fetus responds, or the infant responds. And the reason I say this is because we know that when a fetus acquires a maternal cell here, these maternal cells express this non-inherited maternal antigen. And in this beautiful science paper from 2008, Jeff Mould showed that the fetus specifically regulates against this antigen. So all of these non-maternal antigens, the fetus does not respond to those, becomes tolerant to them. And in the transplant literature, there's this phenomenon known as infectious tolerance. So the idea here is that if you take a mouse, which is strain A, and you induce tolerance against a graft that expresses strain B, you can induce that tolerance. If you then challenge that mouse with strain C, it'll promptly reject that graft, right? It's never seen antigen C before. But if instead you give it a graft that co-expresses B and C together, it'll become tolerant to that graft. And when you subsequently transfer a graft to strain C, it'll tolerate it. So the idea here, this has been deemed infectious tolerance or cross tolerance, depending on where you look. The idea here is that when you experience a neoantigen, in the context of an antigen you're already tolerant to, you're more likely to become tolerant to that neoantigen. There are two proposed mechanisms for this, and they both rely on Tregs. So the first is that you have an antigen-specific Treg here that's seeing antigen A. It's producing IL-10, TGF-beta, IL-35. Any other new effector that comes into that local environment to respond to antigen B is more likely to become a Treg itself. Alternatively, these Tregs can modulate the maturity status of a dendritic cell, which then subsequently interacts with other T effectors and makes them more likely to become Tregs. So the question is, does exposure to maternal microchimerism produce infectious tolerance against new antigens? And that's something we're super interested in understanding. So we already know that if you take cells from a fetus and you stimulate them with maternal alloantigen, you'll see Treg proliferation, suggesting that they're responding to antigen A. We also know from this bystander effect, in this paper they call it bystander effect, but it's the same idea, that if you take mice who are already exposed to NEMA and immunized with tetanus toxoid, you put them into a new mouse and you co-inject tetanus toxoid in the presence of maternal antigen, that maternal antigen will suppress the response against tetanus toxoid. So what we really want to understand is whether this is happening in our babies and our cohorts. And what you might have recognized from this potential model is that it's not malaria specific, right? It suggests that if these babies are acquiring maternal cells, those maternal cells may have the capacity to affect immune responses against a variety of neoantigens that that infant is exposed to. So when we went back to Tanzania and we just said, our babies who had less malarial fever, do they also have less fever in general? We found that these babies who had detectable microchimerism were also getting half as many fevers in general during their infancy. And we don't know what these fevers are from, if they're infections, or if these people are just somehow more tolerant immunologically, they just have a different phenotype. It's possible that these babies, the reason they have all this microchimerism is because they're more tolerant, so they're more likely to accept their microchimerism, and that these maternal cells have nothing to do with the measurement that we're looking at here. So what we also plan to do is set up these really interesting co-culture experiments in which we take cord blood and we add in varying amounts of maternal cells and then stimulate with a neoantigen to see if that dose response of maternal cell or antigen modifies the way that that cord blood will respond to a neoantigen. The other three projects that we're working on to really get at this question are the following. So we're looking at whether maternal microchimerism predicts non-malarial infection in Mali and Uganda. We're looking at whether it predicts BCG immune responses, so looking at vaccine responses. BCG is very interesting to us because it's given on day 1 of life. So if you want to hypothesize that these maternal cells are going to affect anything, we think this will be a really interesting place to look. So this is in our HIV exposed and unexposed cohort. And then with Lisa, we've all met during the introduction, we're looking at whether or not these maternal cells influence the viral reservoir in HIV-infected children, and that's a project we're hopefully just starting. So this is my lab right now. This is Young-Ho and John, who you already met. Christina is based in South Africa at University of Cape Town. Netta and Jacqueline are my 2 incredibly talented undergrads who've been in the lab for about a year and just working super hard on dry blood spot extraction and have a paper they're almost finished with. Netta is now, as her thesis project, is going to go into all of our cord bloods where we can't mark the maternal cells with an antibody and sort, do that bulk sorting and then the qPCR within those subsets. And then Jacqueline, as her thesis project, is looking at all of our pregnant women who have placental malaria, in which we've looked at maternal microchimerism. She's going back and asking the question about fetal microchimerism. So are these women acquiring more fetal cells during their pregnancy? Kelsey is a maternal fetal medicine fellow who's joining the lab in January when she's back from maternity leave, and she'll be looking at preeclampsia and gestational diabetes and looking at fetal microchimerism within that context. So my fellowship work was supported by the Thrasher Research Fund. We've had ongoing support via a variety of mechanisms from the University of Washington and Seattle Children's. All of the non-malarial work is supported by my KOA from NIAID, and the malaria work is supported by a career award from Burroughs Wellcome. The HIV work is supported from a UW CFAR and then from an NIH R01 to Lisa. So in acknowledgement, I think the thing I really wanted to emphasize here is that none of this work is possible without the participation of these pregnant women and their infants from all of these different cohorts, right? And now we're talking about 5 different cohorts and thousands of pregnant women and their infants that we've interacted with, and I think that's incredibly important to acknowledge. All of the other people that I've told you about along the way, the institutional support, all of my collaborators and those that didn't fit on the slide, and then I think perhaps most importantly, when you do this work, is the incredible support of my family, and it's very hard to not take this personally. Right? As both a daughter and a mother of my own children, I think this is incredibly interesting biology, and these are my sources of fetal microchimerism. But I love this idea. No, I haven't. I love this idea that I'm made of cells that are half my mother's genome and half my father's genome, and then I have this contribution of cells from my children that contain half of my father's genome and a quarter of my in-law's genome also. And I think it really expands this concept of self and really opens up this universe of thinking about really non-Mendelian heritability and all of these interesting follow-up questions. So I'll just leave you with this thought, and I'm happy to take any questions. Lisa?
So, Whitney, I'm wondering if you think that there's an evolutionary advantage to this, or do you think it's a happenstance events, kind of like sludging with malaria parasites in the brain, that there's cells that cross, and then I would imagine that what survives was T-Rex, but I'm just curious what your thoughts are.
I think it's a really interesting question. I think a lot of work has been done on fetal microchimerism, and there's clearly an evolutionary advantage to that. So if a pregnant woman acquires a graft that expresses maternal maternal antigens, then she has a higher pregnancy success rate in subsequent pregnancies as she becomes tolerant to those maternal antigens. I think there's very little data on sort of the evolutionary teleologic explanation for maternal microchimerism, and it could be just a byproduct, a happenstance. But I think it's very interesting because our model is really that this is mostly active. We think this is a lot of active trafficking that's happening, and that suggests to us that it's doing something. And within the context of malaria, I think one point I want to make is that a lot of reproductive biology is done in Caucasian women. And our study from Africa was really the first study looking at microchimerism in a non-Caucasian population. So we don't even really know or understand what normal is in that population. And malaria is obviously one of the largest selectors of human genetics in the entire history of the universe. So we think it's very provocative that we see this interaction between these maternal cells and malaria.
So tolerance induction is good if the disease due to the infection is caused by the immune response and the inflammation. But in the case of subcerebral malaria or encephalitis, tolerance is probably would be like predisposed for that. So did you see any of that?
So we'd like to go back and look in our malaria cohorts. One of the challenges in all of the cohorts we work with and with all many African cohorts is just the lack of diagnostics. So we very often know information like the kid had a fever or they had respiratory distress and they had parasites or they didn't have parasites, but knowing what specific infection they had is really impossible. So we want to try to understand that by looking, for example, at GI infections in which we think tolerance will probably be bad because a lot of the pathology is mediated by the infection itself, versus things where we think the immune system is more likely to be detrimental and tolerance would be helpful. Sorry, we just yeah.
If there are no other questions?
So that made a great comment. So if you look at just the bacterial antigens, so 2 to 4% of all pregnant women will get at least demonstrable chorioamnionitis, which would probably have a similar effect. So that's question number one. And the other two, I guess, I'm not sure I'm asking the same one that Lauren asked, but if you were exposed in utero to a group strep antigen, let's say, would that protect you then as a child with strep or similar?
So to answer your question about the in utero exposure, I think the data are mixed. So within the parasite world, exposure to the parasite antigen seems to predispose principally to a tolerant response rather than to a sensitized response. The same thing happens with, we think happens with CMV, and we think the same thing happens actually with hepatitis B. There's some data on hepatitis B. In malaria, when people have gone back and looked at women who were treated for malaria, some proportion of those babies appear to be sensitized to malaria antigens instead of tolerized. And we don't exactly know what that is, what's predicting that alteration response. It's obviously really interesting to us because if you could shift so that neonate or that the fetus was becoming sensitized to a particular antigen rather than tolerized, that could have real advantage in terms of modifying cellular immunity in that neonate. In terms of chorioamnionitis, we have not looked there, but we're very interested in thinking about other places in which we see that infection and inflammation against the placenta. So we're definitely open to collaborations.
Given the role of tolerance in this, have you looked at pathogens that have evolved to have similar sort of to like self-antigens and the like effects on rates of autoimmune disorders?
So we have not. I think CMV is one pathogen that's quite interesting because one of the CMV antigens looks a lot like a self-antigen, but we haven't really looked at that. Thank you.
So, thank you. We have a little student here.
Thank you.
The mention of the students kind of picking up and you are appreciated. Okay, these spots, the speakers are selected as council officers graduate. So this is actually appreciation for the years of all.
Thank you.
Okay.
Thank you so much. I'm very busy.